Imagine if a simple bread could help detect early signs of diseases without needles, blood tests, or invasive procedures. Hello everyone, I'm here to present our research project titled Development of an Electronic Nose to Estimate Metabolic Syndrome in Dog Rats. This research combines biomedical science, data analysis, and engineering to create a medical device for detecting diseases-related biomarkers non-invasively, specifically through breath.
Hello, my name is Napadin Tong.
My name is Cecilia Heswick. Metabolic syndrome affects metabolic processes and leads to the release of specific volatile organic compounds, or VOCs, in the breath. These VOCs can serve as indicator of physiological changes. Our objective was to develop an eNose system capable of detecting such VOCs in dog breath. But where do the VOCs in our breath come from, and how do they link to our health? To understand this, we need to explain by the pathway. For people with diabetes, when the body can't use glucose for energy, it starts using lipids instead. This process produces ketones like acetone, which accumulate in the bloodstream and are exhaled through the mouth. Normal acetone level in breath should be below 0.6 ppm, but people with diabetes have more than 1.25 ppm. For people with kidney disease, when kidneys are not working well, they can't filter urea properly. Ammonia then accumulates in the blood and is exhaled. People with normal kidney function have an ammonia level in their breath about 300 to 1,500 parts per billion by volume. And for people with breast cancer, when alcohol is consumed, it is metabolized by the liver using the enzyme called alcohol dehydrogenase, turning alcohol into acetaldehyde, a known carcinogen, which is a substance that can cause cancer. Both ethanol and acetaldehyde can accumulate it into the bloodstream and exudate it through mouth and skin. In normal people without breast cancer, their breath should not contain alcohol at all. The electronic nose, or eNose, is a technology that mimics the function of the human nose. It uses sensors to detect volatile organic compounds and provides quantitative results in parts per million. The system consists of two main components, chemical detection and pattern recognition. The objective is to design and calibrate a low-cost electronic nose that can detect alcohol, acetone, and ammonia, and test the eNose performance in identifying alcohol odors emitted by humans and cat breath. I will explain the device function in a step-by-step sequence. Start by opening the valve for the clean air to flush in the chamber and establish a baseline. Once your sensor reading stabilizes, move the breath collector close to the subject exhalation. Close the clean air valve and open the sample valve. Breath is drawn in while the air pump. After a set duration, close the sample valve. Sensor record values in real time. Once the reading stabilizes, clean air is introduced again to reset. We use three gas sensors. BME680 measure temperature, humidity, pressure, and air quality. MQ135 measure ammonia, sulfur, benzene, and carbon dioxide. And MQ138 measure acetone. And we have two different types of sensor, which is P-type and N-type. P-type sensors show an increased signal with higher gas concentration, different from N-type sensor, which show a decreased signal. And we connect each sensor to an analog port, showing above. There are two boards, and this is the circuit design. The device includes a cell container, Arduino microcontroller, circuit boards, LED module, button switch, aerosol chamber, and an air pump to control the flow of breath samples into the sensors. Then we connected the whole box with a container filled with activated carbon and an air valve. And here are the eNose device from the side wheel and top wheel. The program, we mainly use Arduino for coding and Excel to create graphs and analyzing results.
For the experiment, we break it into two parts. The first part is, does mass affect the sensor's value? And for the second part is, can our sensors detect changes in VOC, specifically in alcohol? The first part is, does the mass affect the sensor's value? The experiment consisted of four major parts, and I'm going to break it down to you. The first step is to start the electronic nose to allow the sensors to heat up. We connect the Arduino to its power source and then close the valve, and lastly we turn on the air pump monitor. Our sensors are semiconductive, which means it needs heat to be able to operate. Next, we wait until the data shown on screen to become stable before we proceed to the next step. As for the second step, we breathe through the aerosol chamber for 15 seconds. Right after 15 seconds, we immediately close the valve to ensure that we have the same volume for every experiment. We position the aerosol chamber near the snout or near the breathing area without fully covering it to prevent discomfort. You might be wondering why we breathe in for 15 seconds. That is to make sure that every air in the container is sample air. For the third step, we turn off the monitor and close the valve. Then we wait for about 90 seconds to collect the data. And lastly, we turn on the monitor and open the valve to reset the sensors. Next, I'm going to show you the process of analyzing the data. We use the resistance of each sensors and graphed it in the same time duration to see the patterns. You can see that in the beginning, the graph is stable, so that will be a baseline. Next, we breathe into it for 15 seconds before closing it again. We wait for another 90 seconds before opening the valve and reset the sensors. You can see that each graph has its own highest peak, and we use its 10 highest peak and average it to fill in the chart. In the second part, we experiment on whether our sensors can differentiate between a normal breath and an alcohol-influenced breath. This method we use is basically the same as part 1, but we include a step in which we use a mouthwash before breathing in. The mouthwash that we use contains 21.6% of alcohol, and we use an appropriate amount that people use daily, which is 20 milliliters, to prevent harm. The result of the experiment. First is the table that shows the average resistance of 10 highest point for the cats, and this is its graph. Next is also the 10 highest point for volunteer 1 and 2, and this is their graph. Lastly is also the 10 average resistance of each sensors, but this time we use alcohol, and this is its graph. This is the detailed summarizations of each graph. Overall, the graph suggested that larger body mass can create much more intense change and a faster reaction time. For humans, the MQ138 can goes up to 2.2 ohms, and the BME can dip to 26,000, which means that it causes less change for the cat. Next is the detailed summarizations of part 2. You can see from the fourth graph that the MQ138 increased significantly throughout the durations. This also happens to BME, which dipped drastically throughout the process. You can see the same happening to volunteer number 2. Now, let's discuss. The sensors show distinct reading for different mass. The higher the mass, the more intense the graph becomes. They can also differentiate between different species and different mass of the same species. However, further experiments need to be deducted in order to find the pattern of each reading. As for the second part, the sensors can differentiate between a normal breath and an alcohol-influenced breath, with the MQ138 showing higher peak and the BME dropping lower. In the future, we plan to test on more breath samples to create a much more reliable base for future researchers to use. Next, we plan to compare it to other biomarkers such as blood and urine tests. Lastly, we will find the accuracy and efficiency of our electronic notes. Through our work with affordable MQ and BME sensors, we are building the foundation for low-cost and reliable breath analysis. In the future, this technology could help doctors quickly detect alcohol level, infections, or even early signs of cancers and diabetes.
With high sensitivity and real-time results, our electronic nose aims to make medical analysis accessible and affordable for everyone. Thank you for listening.